HIV is able to enter inside CD4 T cells, which is part of the immune system by binding to the GP120 HIV envelope protein, a protein exposed on the surface of HIV to the CD4 receptor and its co-receptor CCR5, another key receptor of the immune system expressed on the surface of the T cell, a type of white blood cell. Receptors are colored yellow and orange here. They allow the binding between HIV and the participant CD4 T cell to occur. T cells that have a chimeric antigen receptor or CAR-T cells have been engineered to perform better. They are a type of immune-based strategy that expresses anti-HIV proteins on their surface. The engineering process will allow them to have a more robust immune response against HIV. Duo CAR-T cells consist of two CARs that supercharge the T cell's fight against HIV. One of the CARs is based on an antibody that can bind to HIV's envelope protein, and the other CAR is based on CD4, which also binds HIV envelope protein. This is how duo CAR-T cells are engineered. A lentiviral vector carrying the therapeutic duo CAR genes are introduced to the T cell, delivering the dual CAR genes into the DNA of the T cell to enable targeting the HIV envelope protein, so that the CAR genes are now active on the cell surface. This can be done in both CD4 T cells as well as CD8 T cells. The duo CAR design protects T cells from HIV infection and redirects T cells to eliminate cells that contain HIV. Elimination is carried out by the release of cytokines by CD8 T cells. Cytokines are small proteins that clear cells containing HIV, as well as activate CD4 T cells, enabling the immune system to fight off HIV and cells that contain HIV. The dual action of dual CAR-T cells would help to reduce HIV viral loads and prevents loss of CD4 T cells. This would help protect the participant against returning or rebounding virus. Clinical trials are happening to determine whether the anti-HIV dual CAR-T cell therapy is safe and effective at keeping HIV durably suppressed in the absence of antiretroviral therapy.